No, I was born. I guess probably before when but yeah, I suppose I have a short one. I don't know. See, so maybe I have a slide of introduction. For you are. Yes, usually when I talk to students, I decide to show them. I really need see, I guess so. I don't know. I mean, I didn't bring anybody here like. So, where did you do your PhD? In Texas, University of North Texas. In Texas. Right up north. I think that's nobody knows. So. This is a slide that you're looking at, you're including. Yes, yeah, it's the first slide actually, I think. Perfecto, perfecto. So I say a couple of things. You did your PhD in
University of North Texas.
Where is it?
In Denton.
So the undergrad in Pisa?
Yes.
Pisa, PhD in technical. And then you directly became a research scientist in I will say that you had a prolific career.
Oh, well, I'm close to 50, so I should have,
career in science. And now you're the vice president of computation at Los Altos Labs.
Not Los Altos, but solo
At Altos. Sorry. Yes. No. Muy bien. That's more than enough. And then you can detail whatever you want.
I'll say a few things, but a few more things.
Formality at the end of the day. We need to introduce each other.
I know.
Lucas, ¿te acuerdas?
Sí.
Lucas es donde vamos a ir.
Sí.
Y abacanos. 7. Altos. Y los álamos, si sabéis.
Yes.
Los altos.
Los Altos it's a town.
It's a town, but nothing related to research.
No.
I was thinking, what am I confusing?
No, you're right. Los Altos.
Proyectos estratégicos, whatever that is. Just Simone,
Sí.
why does the web say that you're vice president of computation?
Yes.
In here it says vice president kind of AI.
Yeah. Yeah, it's a.
Which way? I say computation.
You say computation. You see it.
Otherwise, hybrid is already complicated enough.
We're all vice president of computation, and that's not the area that I call my
And
slide.
these are the top that you do on top. Oh, no.
Yes.
No, todavía no. Al final no. Ayer fuiste a la bienvenida.
Sí,
Porque, nice to meet you. Ya. Sorry.
¿Qué tal?
Muy bien, ¿viste? Pues al final quedé con mi hija, porque finally I met my daughter to have dinner with her in the same place.
Así.
Because I thought, okay, I originally think I have a 21-year-old daughter and a 2-year-old daughter.
Oh,
So now to have to balance a little bit.
again.
Yeah. So
Yeah, when you say I
agree.
miss my daughter, it was like, oh, I
no,
guess this is the older one.
Yeah, because in Saturday she's going back to the university. She's there for a year. She studies in Girona, so she's kind of far away from here. And yeah,
So it was good.
so it was another good, yeah.
Yeah,
the
no, we were
dynamic,
like the usual
Pictures,
fruit in
styles,
the
yeah, good.
little parts.
That's nice. Okay, good.
He's a professor of chemistry at the university, yeah, and he did his postdoc in the campus. Doesn't brag about it, but he should.
Yeah,
Hola, ¿qué tal? ¿Cómo estás? Pues muy bien. Vamos ahí. Nice to meet you. David, he dicho que estoy enfadado con porque cuando la euro montó aquí la conferencia workshop un día,
We are already
hicimos una cosa.
ready. Yes. It's like Wednesday I went to the
Y llamamos.
they were not expecting the speakers attending the workshop.
Okay.
They were like, didn't research for importance, what they, but no, don't worry. I don't Here's my presentation to the
Y el de IPC es las de
and I have the point
el mismo día desde abajo.
we get quiet.
No.
Nancy Viridiana, Nancy Yes. Yeah.
Together.
Whether not quite.
Yeah, exactly.
Me gusta mucho limpiar.
That you Italia and try to get these advantages. You know, university I have to go with them. I don't want that.
Yes.
It is, in this case I want.
Yeah.
Pretend that How long are you staying?
Resting and the. But next week. One of them, then Tuesday. Yes. Well, like there.
Me cambio, es un poco más.
Feeling the same.
Sí, hola,
We don't have
I don't pero sí, desde que nos está todo el mundo habla. Después
Are you originally from the States?
But I've been in the US,
especial y todo. No, que yo no pensaba así, sí, parece más por eso decía lo de que el tío de telas que parte por el lado.
very good. Actually, putting a special but put this in
You go from hard also.
we can do fancy. Usually it's somewhere and more at the beginning of the summer.
Yo no pensé que We wait for 1 minute or 2.
¿Qué tal, Ricardo?
Muy bien, para este minuto.
Muy bien. Muy bien. Ya se me va la mayor a la uni.
Sí,
Cuarto, ya bien.
A mí no es más que yo.
No,
Yo he terminado. ¿Dónde va?
Irene, no es enfermería. Ya, bueno, sí, ya no le doy la pero así la arrancar súper bien y tengo la posibilidad de segundo gol. Ya estoy a gusto, ya me he hecho aquí a
Es bueno, ¿cómo?
y muy bien. Entonces, sí, sí, no, ya vuelvo a sola, apenas le veo en verano.
Bueno, venga, muy fácil. Well, hello everyone. Thank you for approaching and coming to the seminar. So it is my pleasure to introduce Simone Bianco to all of you. Simone, in a few words, did his undergrad in theoretical physics in Italy, in Pisa. After he moved to the United States, to the university, well, to do his PhD in Texas. And after his PhD, he started a prolific career in science. And nowadays, he is the vice president of computation at Altos Labs. So he will tell us more about his career in a minute, but it is my pleasure to join you.
Okay. Now, here. And, do I need to speak for the Zoom? No?
Actually, maybe, I mean, there is a mic.
There's a microphone. Okay, sure, I don't have to do anything. All right. Okay, thank you again for the introduction and especially for the invitation. I'm very excited to be here. I've heard from Sarah a lot about this place, and I'm very happy. It looks fantastic, and I'm as you know, we're going to be here for the next week or so. So maybe there's a chance to talk to some of you if you are so inclined, and if you like what we do at Altos. I usually every time I talk at universities or where there are students, I put a slide that shows the complexity of the career when you are in an interdisciplinary field, like a lot of us are. I started as a theoretical physicist, and then I moved. I was doing nanomaterial, doing my PhD. And I moved very quickly to the life sciences. I worked in epidemics for a number of years. Then when I went to UC San Francisco, I started working on cancer, and then again on the evolution of viruses. And then I transitioned from the university to the industry. I was at IBM for about a decade, where I went, all the way to be a chief strategist for epidemiology and for high risk, high reward programs in life science. And since 2021, I've been at Altos Labs, where first I was principal investigator and director of computational biology, and now I'm vice president, where I oversee the intersection between artificial intelligence and let's say more classical physics-based and molecular modeling for our problems. And so what are our problems? So Altos Labs is a new biotech, is a was funded in 2021, is the largest startup ever funded with about $4 billion of an initial endowment, mostly philanthropic, which has as an objective to fund a new way of doing medicine that is based on really reversing disease. So the idea is to try to understand what are the causes of the deterioration of cellular health that are at the base of a lot of diseases, so that we can act before to what we call cellular reprogramming. And the idea is, again, to be proactive and increase the resilience of cells so that we can live healthier for longer. Altos we have 3 sites, Redwood City, where I work, San Diego and Cambridge. And we are, let's say, thematically divided into several institutes. I'm part of the Institute of Computation that is directed by Hannah Al-Samad. And in the leadership, there's also Morgan Levin, who's the VP of Biological AI, and Vikash Sabata, who's the VP of Software and Scientific Computing. And we basically oversee various parts of the research agenda. All right, what is Altos and what do we do? As I was saying, so the idea is to be able to restore health to cellular reprogramming. I want to say something before. This is really, so I'm going to talk about something that is unpublished. It's the first time that I talk about it, so any omission is my own, of course. But also, as you can see, the story shapes just like research shapes. And so it's not as polished as you would imagine something that has been already published. But let's go back to the presentation. So about 20 years ago, Shinya Yamanaka and his colleague Takahashi discovered that if you express 4 specific transcription factors, 4 genes called OCT4, SOX2, KLF, and MYC, and more colloquially called the Yamanaka factors, you can take any cell, for example, an old cell, and make it become what is called an induced pluripotent stem cell. And these stem cells can, of course, become any other kind of cell. They were awarded the Nobel Prize in 2012 for this fantastic discovery. And this was really the start of a new way of looking at regenerative medicine that starts from taking cells, for example, from skin, and then reprogram them to become pluripotent, and then use them, for example, to regenerate organs, restore tissue functionality, and so forth. The problem was that it doesn't actually work. So if you put it in an animal, you take, for example, a skin cell, you reprogram it, and you make it become a muscle cell, and then you transplant it in an animal, there's a high chance that it will generate multi-tissue growth similar to tumors called the teratomas. And so although it is used in various situations, it's still therapeutically very difficult to really have something that is based on this technique because of the high prevalence of these tumors. And so what we discovered a few years ago was that if you actually stop the process of inducing these pluripotent stem cells, so you start from, for example, a skin cell, induce the Yamanaka factors, but then you stop before the cell loses what is called its somatic identity, so it's still a skin cell, you can prove that all its age-associated hallmarks have been erased. So the cell is practically rejuvenated, although it has not become a stem cell. So this is called partial cellular reprogramming. It was developed by Juan Carlos Espesua Belmonte, who is one of our directors. And in this landmark paper, they've shown that you essentially can rejuvenate cells by inducing the Yamanaka factor just partially. And if you take, for example, an in vivo model like a mouse that has progeria, which is an accelerated aging disease, you can really rejuvenate it practically. For all practical purposes, the animal is rejuvenated. Its life span is extended. and so forth. And so this was really the start of ATOS, right? So you have something that practically you can use to rejuvenate animals. You restore health and resilience, but you don't know anything about it, right? We don't know how it works. We don't know why it works. We don't know we barely know the mechanisms. And so as computational scientists and as physicists being interested in understanding the way life works, this is a perfect opportunity to use some of our modeling tools and some of our background to explain and then try to control some of these mechanisms with the objective of course, of developing therapies that then can help people live healthier. All right, so this is the introduction. Part of what was seen in this study was that also the genome of these animals was kind of modified, let's say, in a sense, and was restored to a more youthful genome. I'm going to explain what that means in a second, but keep this in mind. So the first thing that I want to say, again, as part of this introduction, is that although we're made of 30 trillion cells, and they are all right now reproducing and sustaining us really, all of the different kind of cells that we have in our body have the same genetic code. And in fact, this points to the fact that our genetic code, our DNA, represents an incomplete set of information with respect to our appearance, right? So, although the information regarding the color of my eyes or my height and so forth is encoded in DNA, knowing our DNA doesn't actually tell me what my appearance is going to be. What is going to tell me what my appearance is going to be is the epigenome, which is the realization of the genome through activation of specific gene patterns, right? And so this is called epigenetic patterning, and is done practically through control of the expression of genes in a very dynamic way. Let me introduce you to this, if you're not familiar, to this biological object called chromatin, which is basically the backbone of our chromosomes, and is composed of the DNA and a number of other proteins. The most important ones being histones. Histones are you can consider them like wheels around which the DNA wraps. Histones can be chemically modified to various chemical and biochemical means, and can change the way and the tightness of the DNA wrapping around them, to the point that if the DNA is wrapped very tight, you can form clusters of these histones that, for practical purposes, prevent transcription factors from binding and expressing genes. So how do you actually control gene expression? Well, you engineer, if you are, Deus ex machina, you engineer a system that makes this the machinery to transcribe genes inaccessible. This is what histones do when they clump together, and vice versa. When you want a gene to be expressed, then you unravel this clump, and you leave the DNA open for transcription factors to bind and then transcribe the genes. And again, this is a very dynamic process that is governed by these epigenetic factors. So what was found in our work and in work of others was that during rejuvenation, also the epigenome is restored to its youthful appearance. Why? Well, as we age, the epigenome changes just due to stochastic accumulation of errors, due to drift, due to disease. The epigenome, so the expression of the patterning of the expression of genes that we have when we're young, gets, using a colloquial term, screwed up as time passes, so that the program that is encoded initially that maintains our cells healthy is now has now changed to a program that doesn't work just as well. And so we have accumulation of error, there's no DNA repair, and as our cells replicate, they propagate these errors, so that when we age, we kind of get the flaws that are related, all the maladies that are related with old age. This is, of course, only part of our aging process, but it's a big part of our aging process. So what we showed was that if you use the Yamanaka factors, you restore the epigenome of a cell to the epigenome of a young cell, which allows it to work better. And so, why? So how does this happen? So, in order to be able to control this mechanism, we want to understand how this happens, and this is exactly what I'm going to try and show you today. So the first thing that we need to so these were the main questions. So how is rejuvenation possible? What are the processes that ensure the transmission of the program from one cell to the other, that then we have to tweak so that the program can be restored and the cell can again act as it was a youthful cell. And so again, the idea behind this is really that if we are able to restore the health of the cell, we are able to restore the health of the organism. Of course, we have not been able to prove that in vivo in humans, but we have been able to show that in animals. So we know that something like this happens and that it works. All right. As physicists, what do we do? Well, the typical spherical cow. So I'm going to try and simplify things. Let's take chromatin. Chromatin is a polymer. It's something we know a lot about and we can deal with from a theoretical perspective. And let's consider a chromatin chain composed of several histones. Each of these colored spheres is a histone. And let's imagine that a histone that has been modified, for example, like great. All right, for example, like the ones that we saw that were clumped together is now indicated with a 1 and everything else is at 0. This is a digital system, a digital signal, and we can analyze it with digital signal processing. I'm going to do that in a little bit. But let's consider something like this. Okay, what we're trying to understand is how this signal is maintained across cell division. So as I'm talking now, my 30 trillion cells are replicating. As they are replicating, they are transmitting the genetic code, the epigenetic code, to their daughter cells so that they maintain functionality. A skin cell needs to remain a skin cell. A muscle cell needs to remain a muscle cell. And so it's important that the epigenetic signal, which again, it's related to which genes and how they are expressed, and so how our cells work, is transmitted with a certain fidelity. As I was saying before, so errors in this process are correlated with aging. So this is what we want to try and understand, so that we want to try and modulate for reprogramming and rejuvenation. How does this happen in practice? So imagine that this is the genetic code of the parent cell, and it gets divided during cell replication into the mother cell and the daughter cell. What happens is that each cell gets exactly half the histones. So it gets exactly half the signal that was in the mother cell. And the histones that are missing now get replaced by naive histones that have not been modified. And so we are in a situation after cell division that, well, randomly, 50% of the signal is diluted. 50% is given to the mother, 50% is given to the daughter, and now the problem is signal reconstruction. How is the cell capable of going from this pattern to this pattern, and then now back to that pattern, right? To this functioning pattern now. How does it do it? So efficiently that it maintains a complex organism like a human working with no issue for hundreds of thousands of generations. All right, let's look at a little bit at some observations. So first, this process here, the process of going from an unmodified part of an unmodified histone, an unmodified part of the chromatin to a modified part of the chromatin that ensures clamping, for example, is mediated by enzymes that catalyze the reaction. And it has been shown that these enzymes preferentially attach to areas of high density. So areas like this one, where there are clamps, where histones are modified, will attract other enzymes. So if we have a distribution of enzymes out here, they will preferentially bind here. So this is one observation, and this has been seen in various ways experimentally. The second is that chromatin being a polymer in 3D, so we imagine chromatin, usually the usual characterization of a chromatin is like you see these strands crossing. The reality is that chromatin in the nucleus is actually like a bowl of spaghetti. So it's all mixed together and clumped together. And so it will create these loops, and it will do it using, maximizing information entropy, really. And so knowing that these enzymes will bind preferentially where histones have been modified creates the opportunity during loops to spread the pattern. This is actually known in polymer chemistry and in polymer physics. And I'm going to show you what is the law that actually governs this. But for our purposes right now, what we need to know is the fact that chromatin creates these loops, and then these loops allow histones that might be further away in linear space to get close together and facilitate the long-range spread of marking. So this is the second mechanism that we're going to consider. And so to summarize, a simple model of transmission of genetic information goes through 3 phases. The first phase is the phase of the genetic signal being transmitted, and then the second phase is the cell separation, and the third phase is the restoration of the signal that happens to these mechanisms. At least this is what we hypothesize and what we have seen in the experiment. The localization of these enzymes, the formation of these loops that bring together parts that are far away of the chromatin, and then the spread of the mark to the presence of the enzymes. And so let's go back to our model. Again, so we have this chromatin chain, we have these enzymes that are being transmitted. Half of them stay with the mother, half of them, the histones, half of them stay with the mother, half of them go with the daughter. They get replaced by these naive histones that are not modified. So if I have now a chromatin strand, how do I calculate the probability that if I have a histone that is unmodified, it will be modified? Well, I'm going to just join the probability starting from the mechanisms I have given you before. So first is the probability that a modified node will make contact with a modified node, because the modified node will have the enzymes. And then the second is the probability that the enzyme is actually there. We can calculate this. The first comes from, again, from polymer physics, as I was saying. It's a very well-known law of probability of contact in a linear chain that tells me that any two beads in this polymer chain can get in contact with an inverse power law that scales with a certain exponent that has been measured in human cells as a function of the linear separation along the chain. And this really, I don't have the time to go into it, but this is really, it comes from the maximization of entropy for the formation of various loops. And the second is the probability of binding. This is, again, is a let's say, a known effect. It describes the probability that the enzyme will bind to its substrate, and it's driven by a Hill function where the coefficients can be estimated from experiments. And what we add here is a bias term that we are going to use later to try to understand if we experimentally can change the process of binding, for example, of these enzymes to the substrate. And this is going to be useful later. But so again, to summarize, the probability that an unmodified histone will be modified is going to be given by this law, where PL and PE are the two equations that I showed before, that describe, again, these two mechanisms, the looping that brings together two monomers and the colocalization of the enzymes. All right, I think now we have everything. Is there any question about the theoretical model? It's pretty simple, really. I mean, the fact that if it works, I'm not going to tell you if it works or not. The fact that complex biology can be described by these coarse-grained models, to me, is incredibly revealing of the power of physics and maybe the simplicity of us as organisms. All right, so now that we have this, we can actually do simulations. We can simulate a polymer very efficiently using, for example, a continuous time Markov chain. So we can simulate chromatin, we can simulate histone spreads, and then we can try and see if this maps our data to try to figure out if the mechanism that we have identified are actually the right mechanisms. The idea here is that we take, now in the simulations I'm going to show you now, we're just going to take let's say, a mock genome, and then I'm going to show you a real genome of humans and see if this actually works. We start, in all the next plots, time is going to be on the y-axis in generations. Every generation is the replication of a cell. Every time a cell replicates, again, it will lose half of this signal, and then I will use only those two mechanisms to try to reconstruct it. And then I'm going to go across generations to see if this is stable or not. Again, a young cell is capable of maintaining its stable signal so that it maintains life, right? And this changes as we get older. And again, reprogramming resets this process. Okay, so these are some of the simulations. Again, so time is on the y-axis from 0 to 100 generations. And what we have done here is that we have marked, on the x-axis is the chromatin. This is one epigenetic mark. Each of these little dots is a histone that has been modified, and histones here are unmodified. This is completely stochastic. Any histone can actually be modified according to that equation that I showed you before. And just applying these two mechanisms, what we're going to try and understand is if it is enough to have those two to maintain the signal. And the simulations show that although there is noise, it is those two mechanisms are sufficient to maintain the epigenetic signal. And this is true in a variety of configurations. So it's independent really of the closeness of two marks. And again, remember your genetic code, your chromatin is incredibly complex inside the nucleus. I have a simulation in another presentation that we did that shows how complex it is, and I should have put it here. And it doesn't really matter how many you put, it's really works really well. Not only that, if you remove one of those, so here we remove the density dependence, so the binding of the enzymes, and here remove the polymer looping, you see that this breaks very quickly. So this is, again, the signal at the beginning, and you see here, without the density dependence, it just spreads everywhere. So everything becomes marked, and here nothing is marked. And so this is for us is a good confirmation that this is a minimal mechanism that describes a minimal model for chromatin, for epigenetic memory and epigenetic maintenance. We can go a little further and try to see if it works with data. Now, this is real data. I wanted to focus on the dark blue and the dark red curves. These are human dermal fibroblasts from an individual, from a child of 9 years old, and this is for a specific histone mark that is associated with aging. What you're seeing here is the level of the epigenetic mark. So each of these dots, each of these parts is basically one of the dark things here. And this is after 100 generations. What we have done really is we have taken the cells from the patient, we have grown them in a dish for 100 generations, then we have done the epigenetic sequencing, then we have taken the epigenome at the beginning, we've used that as an initial condition for our simulation, then we ran the simulation. Now the red and the blue are comparing what we see in the real case versus the simulation. And there is a pretty good agreement between the simulation and the data. This agreement can be seen as we change the age of the subject. This is for a different chromosome. And as you can see, it's pretty stable. There are minor fluctuations, but what this is telling me is that this very simple model with these two very simple mechanisms represents the process of epigenetic memory and epigenetic maintenance. And so we can now try and do the next step and try to understand what is reprogramming, what is rejuvenation. All right, one more example. This is for human umbilical cord embryonic cells. These are the ones that get donated, for example, when a woman delivers. And here again, we have a pretty good agreement between the data and the simulations. And I have some, aggregated data here that shows something very interesting. So this is the correlation between the simulation and the data. Shows a very interesting trend that you could have anticipated, that as we age, the epigenome changes. So the model is not capable of correlating as well with the signals telling us that really aging shows a drift in the epigenome that then we hope rejuvenation will reset. All right. I have 10 more minutes. I can tell you a little bit more. All right. So now that we know, is there any question about the data? No. Yeah. Now that we know that the model works, right? Or at least we have an idea that this simple model describes the mechanisms of epigenetic memory. Let's try to understand what happens during disease and what we can do in reprogramming and rejuvenation. And so remember this equation. So this was the equation, excuse me, that showed the binding of the histone modifying enzymes to the histones. And so this is the density, really. This describes the density, the local density of the monomers. And this is the probability of binding. And so what we have added here is a biasing term that we can interpret in the following way. If we had a mechanism, for example, of recruiting or moving away some of these enzymes, we could, in a sense, bias the process of reconstruction of the memory, right? So as for example, we are moving towards aging, where we lose the signal, right? We could use this term, at least theoretically, to recruit more of these enzymes where we want them to be, for example, where the youthful epigenome used to be, and revert the epigenome from old to young. And so the bias, as I was saying, means moving catalytic enzymes. This method is called methyltransferase to a specific site. One more piece of information is that OSKM, which are known as pioneering factor, are actually known to recruit methyltransferases to binding sites. So this gives us one more piece of information, let's say, that might help us understand what happens during reprogramming. So transcription factors go in, they bind to specific sites, they recruit these methyltransferases, which are make histone modifications more likely, and so they tend to change the epigenome. So a possible mechanism of reprogramming linked to the transcription factor may be that there was always, let's say, a coevolution of this transcription factor with chromatin that allows them to reconstruct the epigenome to a youthful one. So anytime we use the Yamanaka factor, this fourth transcription factor, we are resetting the epigenome because they were evolved to bind to places where the young epigenome was supposed to be. This is still a theory, but there's good evidence that that is the case. Let's see if this works. So first thing first, let's consider a random initial chromatin state, and let's bias the signal. So I'm adding just random bias along the chromatin chain, and as predicted, what I'm seeing, even with very weak binding, very weak bias, I see emergence of new signals. This might also be, which is a spontaneous symmetry breaking, might also be an explanation of how our epigenome has started. So just by random creation of these marks and then random selection of the genetic signals that were more fit, it's possible that our own epigenome has been formed. The second thing is that now let's take a single part of the chromatin, and let's give a more clear bias here. Then we see a very clear pattern forming, and this is pattern formation. So we start from nothing, and then we get to a pattern. We can do this in various ways. So for example, we start with a signal here, then I bias to get to this point. And again, I'm just using those two mechanisms. I'm not doing anything else. And I can create now a different epigenetic signal. And so again, this goes towards what we want to do. This is maybe what these reprogramming factors do. We can do various sensitivity analysis for the length of the signal, where we want to apply it, and we can, of course, build a large spectrum of potential cases that now we can use to simulate what we want. So let's go to reprogramming. Let's imagine that this is a healthy baseline. Again, this is time, and this is this is the chromatin, and this is where the signal is. And let's imagine that due to stress or to disease, my healthy state gets modified. Now, because of the stability that is dictated by these mechanistic rules that apply in general, not just to reprogramming. If this is, for example, if this change is driven by a disease, then it's going to be stable. This is aging induced by a disease, right? So our healthy cells, which had this specific signal, now express this different signal here that might not be healthy again. And so let's apply a little bit of reprogramming. So let's assume that we know where this is, and let's apply a little bit of a perturbation here that tries to move the signal back. And so with a little bit of reprogramming, we get what is called insufficient reprogramming. This has been observed as well in vivo. So cells tend to have these patterns that get modified by transcription factors, but they are not necessarily enough. We are not able to naturally, stochastically rejuvenate cells. Let's add a little bit more. And so here we see partial reconstruction of the signal, and this is what we can call partial reprogramming. So just adding the transcription factors a little bit more allows to get the original signal. And so depending on how large this is, we can say, well, this is a successful reprogramming experiment where we have where the animal is again healthy. The cell is again healthy, is expressing what it's supposed to express. And we can go as far as doing full reprogramming, where we, for example, as I was saying before, we induce formation of new patterns that might be associated with a different cell type. And so this kind of signal might be the one that we use to go, for example, from skin to induce pluripotent stem cell and then back to a different kind of cell. And so let me conclude by, I don't have data about this. And the reason for that is, so I don't have data about this. And the reason, well, actually, okay, I do have data about this. The reason I cannot show it is because being a biotech, we first need to understand if this works therapeutically. And then we are going to, of course, try to, but this, all that I'm saying is a paper that is under review in Science. So to conclude, so what I tried to convince you, and I hope I convinced you of, was that really simple physics is capable of capturing deep biological features of epigenetic memory and even very complex therapeutic like reprogramming and rejuvenation. Two very simple biophysical mechanisms, the loop formation and spreading and the preferential binding of the enzymes are sufficient to explain the epigenetic signal, the persistent epigenetic system, and can be used for therapeutic purposes to impose a new epigenetic signal, going to epigenetic engineering and reprogramming. Not only that, but so these two mechanisms, in my personal opinion, are behind the emergence of epigenetic signals. So they go back to the evolution of our genetic code as we have it today, and the evolution of course, multicellular, single-cellular and multicellular life. Epigenetic signals are typical of multicellular life, but they're also seen in some unicellular organisms. And the idea that, you know, biasing the signal can reconstruct a healthy epigenome goes to a potential for the coevolution of transcription factors like the Yamanaka factors and chromatin as an explanation of aging and the potential mechanisms for reprogramming. All right, so being vice president means that you take credit for a lot of things that other people do. And so this is mainly the work of Eric and William, who are very talented scientists in my group. Eric was with me at IBM, and Sarah knows him well. William is from Caltech, and the co-advising of Carlos Lopez, Anna Samad, and the engineering work of Brian Spengler. Most of the experimental data was acquired in the labs of Wolf Reich and Ken Raj in Cambridge, and it was mainly the work of Ariane and Jie-Shung. And let me stop here. I think 45 minutes, as I promised, and I will entertain any question.
Great. Thank you so much, Simone. So I don't know if you guys have questions.
Yes, please.
That was very interesting. I have lots of questions, but one would be, so I guess the simple mechanism is very convincing, no, on providing this robustness, no, to keeping these fingerprints from like epigenomic modifications. However, these four transcription factors seem to be involved in something that's not only related to aging, but the actual differentiation,
Yes,
which has to be related with how different cells become, no, as they specialize. And then, for example, if we think of an embryo,
yes,
what we have is like the opposite of robustness, because in a very short span of time, cells are able to lose all the memory of where they were coming from and become something very, very different.
yes.
That depends on boundary conditions, I guess, and all sorts of other things. So can the model also be used beyond this like a stochasticity term, or maybe that's the key to everything, but can we explain such different things using a simple model like this, as for allowing the flexibility that cells actually get to have in short periods of time?
It's a fantastic question. I think the part of the answer is that although these mechanisms are sufficient, they're not the only ones, right? There's a lot that goes on in the genome that ensures error corrections and repair and so forth, that ensures that the signal is stable across generations, right? And so I wouldn't go as far as saying that this is something that you can use as is. But in my mind, these two mechanisms needs to be part of the inheritance of the epigenome. And so I'd say that in principle, there's nothing that if you change the assumptions and you are in a different set of assumptions, that you could use the same model to try and model some of these other. Now, this is for histone. If you go to DNA methylation, this already doesn't apply as is, right? And so it's a in my mind is a very good, very simple model that allows to captures what we see in the experiments, but I don't think it applies in general as is, right? You need to probably change a few things. I don't know, maybe unsatisfactory answer.
It's very difficult anyway.
It is very difficult, yes, but it's very interesting because at the beginning of the life of the embryo, there are so many things that happen, right? So there's an epigenetic the reset, complete epigenetic reset, right? And that's very poorly understood. The fact is that developmental biology, which is a field where we worked for a long time, is still not, a lot of things are still very poorly understood. So, which makes it exciting. You said you have many questions, so please
take turns.
No, just to continue on that line, you mentioned that this is not the only mechanism that ensure fidelity. The question is to what extent these are, what is the contribution of these? You have two factors. One of them is the preferential binding of the enzymes, the modifying enzyme to an already modified histone.
Yes.
And you showed at the beginning a graph that shows this preferential binding.
Yes.
And you can infer a little bit, slightly, the magnitude of that preferential binding and see if that corresponds to the probability that you had to put into your model in order
Yes.
to ensure this fidelity. And
Absolutely.
if you do it, there is correspondence?
There is correspondence, yes. Well, I mean, within a toy model, with, I think this model in total is 6 parameters, right? And some of them are, they are known. It's not very difficult to make it fit. But what we did when we did the data, when we went to the data, we fixed the parameters and we didn't change them for all of the simulations. And to me, that was surprising, right? So we extracted them from those plots, and then we used them for the 9 years old and the 28 years old and the 70 years old, and it was still. And so that gives me some confidence. Thank you for the question.
I have a very,
Yes, that's all.
I don't know, naive question. I don't know how to say it, but a question from the streets, let's say.
Sure.
Do you think there is any hope that we're going to get to see some pill or something that it's going to help us to rejuvenate ourselves in our lifespan? Or
My personal opinion, which does not reflect the views of Altos, is yes. Actually, I mean, I wish I could tell you, but I've seen things that are unbelievable. And so I'm very, very hopeful. And the things we published are mind-blowing. I mean, the fact that you can take a mouse. I mean, there was a recent paper by another fellow, should I say Spanish? I mean, in the Basque Country, can I say Spanish?
sí.
Yeah.
No, you cannot.
No, I cannot.
I was not understanding your question.
That showed how this works in muscles. And you can see these mice recovering the fitness of very young mice. So these very old mice recovering the fitness of very young mice. So I think it was a Nature paper just a few months ago. And this is a Pura Munoz. She's another investigator at Altus that led that. So I think now how close are we? I cannot tell, but I feel that we are going to see something that is going to hopefully be revolutionary. I really hope.
So when you said that your views do not reflect those of Altos, that's a sentence that you have to say, or do you mean that Altos doesn't believe that there's going to be a pill?
It's so funny because every time I do anything, I have to say that my views don't. I just.
Okay.
It's so funny because I'm giving a talk. I was in a meeting I organized a workshop for the National Academy of Science of the ethics of publishing AI in the life science. And then we wrote a companion paper, which we sent to Science. And so when I sent it to my legal, they said, where is the disclaimer? And I said, which disclaimer? They said, the disclaimer that these are your views. I said, well, it's a paper. And yet you have to say sometimes that these are my views.
So there is hope.
There is hope,
what I
yes.
wanted to conclude. So thank you.
I would say similar to the Aitzol, more like naive from the street, let's say. So you're talking about rejuvenation and then therapeutics. So here the idea is that the process is common for every cell, but for because different disease affect different cells, and the way in which the muscle gets older, then it's different from the disease that affected muscle, for instance, right? So is this a mechanism and the therapeutics then will be tuned for different disease, different cell type and so on, or is a general mechanism that then you have the one single pill that fix everything, no matter what is the disease or if you're just getting older?
This is a great question. There are two answers. The first is kind of obvious. I do feel that the mechanism will need to be adapted. Sorry, the therapy will need to be adapted. But the mechanism, so OSKM work in every cell. they work in muscle cell, in neurons, they work everywhere. And in, mitotic and post-mitotic cells, they work everywhere.